This is Mohan Wani, and I'm very glad to introduce Dr. Kaveri. Dr. Kaveri is currently working as a director, CNRS, Embassy of France, New Delhi, and he's also a director of research in SAM, Paris. And I'm very glad to know that he is my professional brother. He is also trained as a veterinary veterinary physician. He has done BVSc and MVSc from India, and he has done PhD at Pasteur Institute, Paris. And after that, he has done his postdoctoral research training in the University of California, San Diego, USA. The research interest of Dr. Kaveri is immunopathology and immunotherapy, with a special focus on autoimmune and inflammatory diseases. Dr. Kaveri has published more than 250 research articles in well-reputed journals, including several papers in New England Journal of Medicine, Blood, PNS, Nature Reviews, Rheumatology, and I'm happy to tell that he has got more than 20,000 citations for his publication. And today he is going to talk on T regulatory cells. Are they defective in autoimmune disease? And this area is also of my interest. We also work on T regulatory cells. So I welcome Dr. Kaveri, and I request you to please present your talk.
Thank you very much, Mohan Mani, my professional brother, veterinarian. It's nice to see. Do you see my slides?
No,
sir.
No,
You need to share.
Well, I'll share again my screen and come back to voila. I share my screen. And
it was visible, now it is not.
Yeah. If you allow me, yeah. How about now?
Yeah, sure.
Do you see
Yeah,
that?
now, yes.
Yeah.
So now we can see the slide. Yeah.
Do you see full screen?
Yes.
All right. Okay. Thank you very much. And I think we have gone past at least, I don't know how much, quite a lot beyond our schedule. We have 20 minutes past the what you had given us the time, more than 2 hours. So I'll try to make it as brief and as less boring as possible. So before I start, I would like to thank my colleagues and students present, past, etc. And I have indicated particularly my Indian students who came to the lab in Paris, and many of them have come back to India. Some of them have moved on, and I would like to thank all of them. I also would like to thank my colleagues, clinicians, because many of the questions that we ask in the lab are highly clinically oriented, and therefore I thank all my clinician colleagues in France and elsewhere. And as you can recognize, in India, I work with some clinicians and non-clinicians, some groups in Bangalore, IISc, and in Mumbai, and also Veer Singh Negi, that Chandrasekhar knows very well. He's a good friend of mine. I have been working with him also for quite some time, and elsewhere. So before I'd like to start, I have this little disclaimer. See, as this I just put now, a few minutes ago. As I was listening to Girdhari, Rupesh, and Sunil, I was under the impression that I'm watching a movie like this, the James Bond movie or a Fast and Furious movie. These young people are amazing. The way they are doing research these days is, I'm really impressed. They're fantastic. And they were going such a speed, I mean, with the results, not just the speaking, but the results and their observations, their ideas. I felt I was sitting in a movie like that. But then I may have to disappoint you, because my story is more like this. Take it easy, okay? You relax and sit back, and then you may also fall asleep, but try to control your sleep, okay? So yeah, you should have not given me the last slot, because most people may go to bed after listening to these young people who were so dynamic, and I'm a little less probably dynamic. Okay, so for me, in a very simple-minded manner, when we come across a pathogen, our innate immune system can handle that. And as you all know, we have learned in our high school, But when the innate immune system cannot cope up with such pathogens, either because of their virulence, because of their number, because of their dose, high quantity, then adaptive immune system is called for. In the adaptive immune system, as we heard very elegantly from the last three speakers, we have the presentation of the antigen, as we heard very beautifully from Sunil about the role of the antigen presenting cells that present antigen to the T cells, and the T cells in a very finely orchestrated microenvironment dependent manner, and depending on the nature of the antigen and the cytokines, they polarize into different T cell sets. And the T cells also provide help to the B cells. As we all know, the B cells, we heard beautiful presentation by Rupesh about the role of B cells. And the B cells, the humoral function, as we heard, is to make antibodies, and the antibodies are specific. Let's say the antibodies, specific antibodies bind the antigen, and if the antigen by then has reached the target organ, let's say the lungs, the moment the antigen is recognized by the antibody, a whole series of biochemical reactions begin by, to start with, the antigen combining site, it recognizes the antigen, but the FC portion binds or engages with the FC receptor. Engagement of the FC receptor leads to a various effector mechanisms that we just now heard, including the production of some obnoxious pro-inflammatory cytokines and complement activation, and the role of neutrophils also comes in, and the whole idea is to eliminate the pathogen. And this involves, it comes at a small price, which is inflammation, but this inflammation is good. And this works almost to perfection. But if this is not sufficient enough, then you recruit the T cells. The T cells, as I said, they polarize into different subsets. And depending on the nature of the antigen or the context of the cytokines, these T cells also produce more obnoxious molecules, the cytokines, and the end result is the pathogen clearance. And if the pathogen is, let's say, a virus, or sometimes in cancer, the entire virus infected cell can be lysed by the cytotoxic T cell. So this whole thing happens to perfection, and it's very important. Plus, as we also heard, we have a bonus in that we have both the generation of memory T cells and the memory T cells, and this is B cells and T cells, sorry. And this is the basis of vaccination. We all know that. And all this works to really a perfection. However, sometimes the immune system does not behave, either because of some elements that are missing. The immune system is either completely depressed or compromised, or it is hyperactive, or it is misdirected. And when some of the components of the immune system are missing or defective or completely absent, then we have what is called immunodeficiency. And immunodeficiency is obviously quite fatal. And as I just want to pay my respects to my fellow countryman, Alan Fisher, who is one of the first ones to have demonstrated that you can actually do gene therapy in immunodeficiency to correct the genetic dysfunction or genetic missing gene, to insert that and to correct this particular condition. So we also have an exaggerated immune response, and we all know about that, and that's hypersensitivity or allergic reaction when the immune system goes out of control and overreacts. But then we also sometimes have no infection, but the immune system starts mounting an immune response as aggressive as against a pathogen, and that, for example, if it is directed against pancreas or misdirected, let's say, not against the pathogen, not against a foreign antigen, but against a self-antigen, the pancreas, then you end up in what we call as autoimmunity or autoimmune disease. And you know quite a few examples, and we heard quite a few examples in the last. The whole concept of autoimmunity has been around for a long time. However, the real, in the beginning, when Paul Ehrlich in the last century or even before the last century, they said, Paul Ehrlich said, no, immune system cannot recognize the self. If it recognizes the self, it is a horror autotoxicus, meaning it is disastrous. But then we didn't go beyond that at that point. It took some time before we could really define what an autoimmunity is. And that came in the work of Vitebsky, Rose, and Yvonne Reut with thyroiditis and myasthenia and other conditions. Beautiful work, and that led us to define what autoimmunity is. However, around the same time, as we also heard very beautifully by Girdhari earlier, the beautiful landmark work led us to understand how the immune system prevents autoimmunity. And that involves immune tolerance. And this is very important to study the immune tolerance. And this is something basic. Immune tolerance happens both at the central level and at the peripheral level. At the central level, you have deletion of autoreactive T cell clones in the thymus. We heard that. And of B cell clones, sorry about the mistake, the B cell clones in the bone marrow. Therefore, the self-reactive clones are eliminated right in the center, and you don't have autoimmunity. And in case they escape, even in the periphery, you have deletion mechanism, or you can have ignorance of the clonal ignorance or editing of a receptor. That way, you don't have an immune response against self-antigen. Or sometimes you even have a state of energy, which also ensures tolerance. But equally important is the peripheral regulation or suppression. And in a simplified manner, let's say a classically immune response, as we heard in practically all the three speeches earlier, an antigen presenting cell presents the an antigen, external antigen, let's say, under the context of appropriate co-stimulatory molecules, signal 1, signal 2, we heard about signal 3 also, the T cell gets activated. This is perfect. However, certain antigens, for example, the sperm antigens and then the lens antigens, there is an ignorance. Immunologically, they are ignored. There is a barrier which prevents the activation of the immune system against these particular self-antigens. And therefore, your sperm antigens or lens antigens are not subject to immune aggression, and therefore they are spared, and there is a tolerance at that level. And as I also mentioned, thanks to certain very interesting molecules, these are the inhibitory molecules, which are set off the co-stimulatory, but then these are not stimulatory, rather inhibitory. For example, CTLA4, a very crucial molecule in controlling the immune response. And when that is engaged, you do not have an activation. On the other hand, there's a negative signaling to switch off the inflammation. That's another way of ensuring that there is no activation against the self-antigen. But classically, as we also heard, there is fast ligand-mediated deletion of some autoreactive clones, and that way also the self-reactivity in the periphery is controlled. But what we're going to discuss more today is about one particular cell type, which is called the regulatory T cell of the Treg. And the Treg also ensures both in the center and in the periphery kind of tolerance, and ensures that the T cells do not get unusually activated. We have heard quite a bit about the way it is generated, but the way I look at it in an extremely simple-minded manner is in the thymus. We heard the role of thymus very elegantly by Girdhari, and in the thymus, there's a bunch of T cells that are generated, and the thymic epithelial cells, the medullary thymic epithelial cells, they present all the self-antigen in a context of AIRE, A-I-R-E, that Sunil elegantly showed. In the context of AIRE, the self-antigens are displayed to the T cells. Those T cells that do not bear FOXP3 transcription factor, they are eliminated, because if they are recognizing the self, if they recognize the self, then they are actually deleted, and that's why this is called negative selection. And if they are not self-reactive, they go out and they become very good effector cells, and they're responsible for immunity. So you got that. A T cell which is not recognizing the self goes out, and there's no self-reactivity, and this is a very good effector cell. But sometimes the self-reactive T cells, they escape this particular selection process, apoptosis process, and they can get out. And these are responsible for autoimmunity. This is a classically well understood picture. However, what we have also is a bunch of cells that are carrying a transcription factor called the FOXP3. And even if they recognize the self, they are positively selected. They do recognize the self, they're positively selected, they're let out of the system, they are in the periphery, and they can control autoimmunity, they regulate in the periphery, and that is a very interesting population called regulatory T cells, and we'll hear about that. The very presence of this regulatory T cell is absolutely unquestionable, and that unquestionable beyond doubt proof comes from this fantastic study or fantastic observation where if you have a mutation in the FOX, in the gene that encodes the FOX head protein 3 or FOXP3 in your short arm of the X chromosome, and that means that you do not have appropriate level or correct level of regulatory T cells. If you do not have in the periphery the CD4, CD25 positive regulatory T cell, and these are, of course, FOXP3 negative. Therefore, there is no cell, regulatory T cell, and these young boys, they end up with a disease called IPEX, which is a very unfortunately a very fatal disease, which is completely total immune dysregulation, polyendocrinopathy, enteropathy, which is excellent. That's why it's called IPEX. So, but these are now conventionally called Tregs. But long back in the 70s, there was a lot of work done on a particular subpopulation, and they were called suppressor T cells. And a huge amount of work was done, but at that time, we didn't have appropriate markers. We didn't know how to demonstrate the very existence because we didn't have the technology and the knowledge on the markers. And therefore, the studies went kind of silent during this particular period in the 90s. Then came two beautiful studies, and one of them is that of Sakaguchi. And based on Sakaguchi's work, and we could isolate a population, and that led to fantastic discovery of regulatory T cells and more detailed studies. His experiment was so beautiful and elegant and simple. What he did was he took healthy mouse, took the entire range of T cells, injected into a skid nude mouse, meaning that this particular mouse doesn't have its appropriate immune system, doesn't have an entire repertoire of T or B cells. And that when you re-establish or replenish the entire T cell population in this particular mouse is happy because it can mount an immune response to a challenge afterwards. But if you take the healthy mice, the same healthy mouse, and deplete completely its CD25 positive cells, CD25 depleted T cells, meaning you inject the T cells minus the CD25, then this mouse gets sick. It develops certain number of autoimmune diseases, and in particular, very clearly, IBD has been demonstrated. Meaning, there is a bunch of cells within this healthy population, which is CD25 positive, which is responsible for maintaining the immune control or the effector control. Otherwise, there are self-reactive T cells, and those self-reactive T cells can exaggerate, exacerbate, and they go beyond control, and that's why you develop autoimmune diseases. So a loss of Tregs in this, meaning a break of self-tolerance, meaning autoimmunity, that was a monumental work, because based on that, a whole set of experiments started happening, and many papers got published. And from the time that they were first discovered or first demonstrated, identified in the mid-90s, if you type now on PubMed, you will have approximately 20,000 papers, 20,000 papers on regulatory T cells. 20,000 papers in the next 20 minutes is not going to be easy. So I'll make it really simple, because we are going to talk about essentially Tregs of one set of population. Because in fact, as we heard also very nicely from Rupesh, any cell that has a regulatory function can be called regulatory. For example, regulatory B cells or Bregs. So even among T cells, there's a whole range of cells that have this regulatory function or suppressor function, and they all come under regulatory T cells. But there's one particular cell type, which is the CD4, CD25 FOXP3 positive. And most importantly, that expresses the CTLA4. And as I told, these are FOXP3 positive, and they have immunosuppressor function. But equally important is the plasticity, meaning the depending on the microenvironment, the way they they're flexible, they move around. We heard that in a very detailed manner, so I'll not touch upon that. But mind you, the flexibility or the plasticity of regulatory T cells is a virtue, because one cannot stay blocked in one particular state in the T cell biology. They are highly flexible, and that's why it's most important. So soon, variety of markers were identified that characterized the FOXP3 cells. They were not whole and soul, but then quite a few markers were characteristic, which unfortunately is not the case in Bregs. We heard about that. And whereas in Tregs, we know that they produce IL-10, they produce IL-35, but they also, most importantly, they express CD25 very high level, they express CTLA4 and quite a range of molecules on their surface. And that is the reason we can very easily study them, purify them, and also use them as a immunotherapeutic tool. And so I obviously will not go into the mechanisms of action of each one of these molecules. And if you go through any immunology book, these molecules are very well described. And some of them are identified more recently. For example, TIGIT slightly recently, but then Helios is also identified. Quite a few molecules associated with regulatory T cells have now been identified. Now, coming to the way it functions. So yes, the regulatory T cells have a very interesting regulatory function or suppressor function on a variety of cells, on NK cells, on mast cells, and monocytes, macrophages, dendritic cells, neutrophils, T cells, B cells, as I said already, NK T cells, NK cells and NK T cells. They are really a perfect regulatory or suppressor cells, and that can suppress the function of an entire range of cells relevant for the immune system. But curiously, we observed that they do not necessarily suppress the function of basophils. On the contrary, Jagdish in my group has done a lot of work on that, and with Meenu Sharma, Jagdish, they demonstrated and published in Science Immunology a couple of years ago that the regulatory T cells, the Tregs, on the contrary to all other cells, they actually activate the basophils. The meaning of activation of basophils is subject to a lot of discussion. Why does a Treg enhance the function or activate the T cells, and what is its role in immune system in general? What does it do in allergic conditions, etc., is subject to a lot of speculation, a lot of further studies, but it's a very interesting and original observation that our lab made a couple of years ago. So after we demonstrate this, but then meanwhile, we need to understand how the regulatory T cells in general, they function. Once again, as I said, first of all, if you go into the details of how each cell is controlled by regulatory T cells, it will take several hours to go into the details of how each cell population is controlled. But let me make it simple. The regulatory T cells exert first their function, the suppressive function or regulatory function, thanks to several molecules. First of all, several mechanisms, and one of them is inhibitory cytokines. Either they carry the membrane-bound tethered TGF-beta or secreted they secrete TGF-beta, IL-35, IL-10, as I said, which can suppress the function of effector T cells. They can directly cause apoptosis through granzyme A or B and causing through perforin pore, and thereby ensuring apoptosis of effector T cells. By disrupting the metabolism of the effector T cells, either by deprivation of the cytokine, for example, IL-2 is necessary, as they drink a lot of cytokine that is deprived for effector T cells, or through cyclic AMP-mediated mechanisms, they also can cause dysfunction of metabolic disruption of the effector T cells. Now, Equally important is the effect of regulatory T cells, brown here, on effect on dendritic cells, and how the dendritic cell function is inhibited is an extremely interesting area of research, because we just now heard very clearly, very elegantly from Sunil, the role of regulatory, the dendritic cells, either in, at the crossroad of immune function, either in activation or in tolerance. Both are important. In activation, because they can, stimulate the T helper naive T cells into pro-inflammatory or into a non-inflammatory state. Or more recently, as we also heard, the same dendritic cells, they can drive the T helper cells to polarize into Th17 cells, which can be or into CD25 positive FOXP3 cells. So with with such a background, knowing the importance of the dendritic cells in T cell biology, we asked the question in our lab, what does a Treg do to the dendritic cells? And the answer is yes. We were one of the first ones to show that the regulatory T cells have an inhibitory, sorry about this ulta or upside down dendritic cell, somehow it went down. Meaning probably it is a function, that may be the idea. No, I'm joking. So yes, we demonstrated that regulatory T cells inhibit the dendritic cell activation, suppress the D cell inflammatory cytokines, they block the antigen presenting capacity, reduce the T cell proliferation, and mechanisms are multiple, either surface molecule oriented or T cell or the cytokine dependent. Namita Mishra showed this in a cutting edge paper in 2004. We were probably the second one to show the effect of it. Followed by that, our group published quite a lot on the role of regulatory T cells on the dendritic cells. And meanwhile, of course, this is a very important area where several papers have been published on the effect of Tregs on the dendritic cells. Now, let me come to the autoimmune diseases. Clearly, earlier speakers have very, very well mentioned, particularly Girdhari, on the plasticity. In a healthy situation, when the Tregs are optimal, both in terms of the number and function, they exert a very balanced effect on the entire range of cells, which in turn make sure that the effector T cells are under control. This is the balance. This is most important to maintain immune homeostasis, and therefore cellular homeostasis and immune homeostasis in general in a healthy situation. But an imbalance in this particular state, for example, either in the number or in the function, then the effector T cells, they take an upper hand, and if they are self-reactive, then they cause autoimmunity. And this is associated with, this automatically leads to many therapeutic options. So clearly, if the title of my talk was, are the regulatory T cells defective in an autoimmune disease? The answer is yes. They are, and it has been demonstrated in a variety of animal models, in type 1 diabetes, in arthritis, in multiple sclerosis, EAE model, asthma, as lupus, GVHD, systemic sclerosis, and myasthenia, that either the phenotype or the quantity or the function, this is true for all these diseases. So I can go on explaining about each one in how each disease, what is the defect that happens at the level of regulatory T cells. But it is sufficient to say that in most autoimmune diseases that have been studied, or in all the autoimmune diseases that have been studied in animal models, it is clear that there is a change in the regulatory T cell number, function, and in the phenotype. The same is true in patients, human patients suffering from recurrent relapsing multiple sclerosis, in lupus, in Crohn's disease, Guillain-Barré syndrome, and this is the number of Tregs that goes down. And whereas the suppressive function of the Tregs is defective in recurrent relapsing multiple sclerosis, in type 1 diabetes, in lupus, probably in many conditions. But these are the diseases in which very beautiful clinical studies have demonstrated that the regulatory T cell function or the number is defective. This is the take-home message, very simple. Therefore, this also gives room to envisage or conceive therapeutic strategies aimed at using the regulatory T cells. So let's see how do we try to understand the role of regulatory T cells in the treatment of an autoimmune disease. If you take multiple sclerosis as a prototype of an autoimmune disease, and you all know what multiple sclerosis is, one of the most dreadful autoimmune disease where self-reactive T cells damage certain portions of the central nervous system and thereby leading to paralysis and many other symptoms, many other symptoms. If you take that as a prototype or a typical autoimmune disease, then more than 15 therapeutic strategies are currently available. More than 15, probably more than that, much more than that. And all of them systematically implicate in one way or the other Tregs. I mean, in one way or the other, they enhance either the function or the number of regulatory T cells. I take, for example, type 1 interferon or glatiramer that is used at natalizumab or fingolimab or DMF or anti-CD52 monoclonal antibody, or take any of the therapeutic strategies, including corticosteroids, or for that matter, rapamycin or any other condition. Most of them are associated with the upregulation of regulatory T cells. Why? Even intravenous immunoglobulins. How can I not talk about intravenous immunoglobulins? So those people who have heard me in the past know that my lab has been working on the in the role of IVIG in autoimmune diseases for the last probably 30 odd years and maybe more than that. And right from the beginning, we have been interested in how IVIG or therapeutic immunoglobulins exert their immunoregulatory function in autoimmune diseases. And right from 2007, we got interested in how IVIG can modulate the regulatory T cell function. Sorry if I sound pretentious, we have published a series of papers, first in vitro and then in animal models and in human patients, that IVIG can expand and enhance the function of regulatory T cells. And first we showed that in vitro it can enhance, then we showed in an in vivo model in EAE that it upregulates regulatory T cells. And then we went on to demonstrate with Mohan Madhur and Minu Sharma and Deep Chaitrali and Shiva Oti. Quite a few people were involved in this study. And we showed that not only IVIG upregulates the regulatory T cell function, but it downregulates the Th17 cells in patients with some autoimmune diseases and certainly in animal models. So to show one representative slide, just one representative slide, because this, as you saw, this work on regulatory T cells IVIG probably amounts to 15-20 papers from the group, but just one prototypic, let's say a classical example is in Guillain-Barré disease. Guillain-Barré, as you may know, is associated with an dysregulation of Th1, Th17 and downregulation of Tregs. And we clearly showed that IVIG upregulates over a period of 1 month period, a gradual, slow and steady increase in the number of regulatory T cells. And this is also associated with upregulation of FOXP3 as a marker and Helios as a marker. And we followed it up with many cytokine and other functional aspects. So in brief, most of the therapeutic strategies that are used which in practice currently in autoimmune diseases, in one way or the other, they implicate the regulatory T cells, and IVIG is no exemption. So if you take the animal models once again, you take the same models that I have been showing earlier, type 1, not in animal models, for example, type 1 diabetes, rheumatoid arthritis, multiple sclerosis, or EAE, asthma, lupus, graft versus host disease, or systemic sclerosis. As I said, there is a defect in the number and in the function of regulatory T cells, and the therapies that have been at least tried in animal models, for example, IL-2 or IL-2 MAB complexes, or low-dose IL-2 or rapamycin or adaptive Treg therapy or tregitope therapy. I will probably spend 1 minute on tregitope. See, tregitope is a concept that is quite close to IVIG actually, because tregitope are those motifs. Tregitope, Treg epitope, actually, that's what it is, tregitope. So this tregitope is a concept that was Annie de Groot that put forward this concept, where she and her group have identified particular motifs in the immunoglobulin molecule, which she believes is responsible for upregulation of regulatory T cells. And as IVIG, I showed you a few minutes ago, upregulates, she clearly believes that what upregulates the regulatory T cells in IVIG therapy is actually due to the regular tregitopes, meaning those particular motifs in the immunoglobulin molecule that are responsible. And of course, you have many other therapeutic strategies, for example, anti-TNF, anti-IL-6, or low-dose irradiation also leads to that. So no matter what immunotherapy you are talking about, indirectly or directly, it causes or it leads to an upregulation of regulatory T cells. So there is a direct benefit of using regulatory T cells, and therefore it makes sense to use regulatory T cells, to use them for therapy, obviously. So initial studies, very beautiful studies, they demonstrated that you can make autologous polyclonal max plus fax plus CD425 positive, CD127 low cells, purify them. Either you can use the total Tregs and then expand them, make them more antigen specific, and then, or you can use the naive T6 and then expand them, provide them with mTOR inhibitor, and therefore you can make them more active, and you once again re-stimulate them, expand them, and subject to quality control, and use them. This is what is tried in some of these diseases like type 1 diabetes, lupus, multiple sclerosis, COVID-19, and Crohn's disease. Obviously, we are talking about, let's say, polyclonal population, not very specific. So they might be associated with some of the disadvantages of not being specific. And of course, despite that, these are the clinical trials that some of them have completed, some of them are still going on in type 1 diabetes, lupus, pemphigus, as I said, autoimmune hepatitis, and Crohn's disease. So yes, there is room for polyclonally expanded regulatory T cells. These are all polyclonal. And so you may be thinking, why don't we make it more specific? And that is when the CAR T cells that come into picture. And you all know the principle of generating the chimeric antigen receptor, and you can use that in some conditions, but then you don't want to make every B cell, you don't want to knock off every B cell. That would be like you're using a rituximab. So you design a T cell in such a way that it's specific for the surface receptor of an antigen specific B lymphocyte, so that only a specific antigen, specific B lymphocyte is lysed. But this is in very rudimentary stage. It's way too early to predict anything. But people are already thinking. So the principle would be similar. You generate total Tregs, and then you expand a certain population of the Tregs, and then this is when you insert the Treg master product, meaning this is, you have the group, and then you make a CAR design, make a more specific insertion of a vector, and therefore you make them more specific for your antigen, or at least the B cell that carries the surface immunoglobulin receptor, and expand them and use these particular CAR Tregs and make sure that they meet all the quality control, and then go for therapy. But this is this is not yet. This is probably, it will happen soon. People are thinking seriously, but we are not there yet. I made it a little short and sweet because I know that the time is running fast, and then I was a speaker. I removed quite a few slides because most of the speakers earlier, they covered a lot about regulatory T cells, and they did a much better job than what I could have done, and the young people have done fantastic job. You brought in fantastic speakers, Chandrasekhar, today. So, and Girdhari, thank you both for inviting me. So one last slide, the way I look at the immune system, many people have seen this, but this is the way I always believe, unless I'm wrong, unless I'm proven wrong one day, which may happen. So to me, this is a perfect immune system when all the elements are under control, or under, the good cells are controlled by, good cells control the bad cells, and the good antibodies, like the anti-idiotypic antibodies or regulatory antibodies, or the regulatory B cells control the bad cells. But however, in an autoimmune condition, clinicians know better that it is not just because of an unusual expansion of a clone or an abnormal behavior of one molecule. Soon, the entire immune repertoire goes berserk. Entire immune system goes, the dynamics is completely disturbed, the architecture is disturbed. And what I believe is, whenever you conceive an immunotherapeutic strategy, even including the regulatory T cells, you should make sure that you only come back and reestablish the homeostasis. So even when you conceive regulatory T cell therapy, make sure that you are not suppressing the entire immune system. Therefore, you may want to be as specific, as precise as possible. And this is exactly what Chandrasekhar and the clinicians have been asking. How do we know? When do we have to give? What exactly is the window of treatment? And do we know the status of how we can use such immunoregulatory or immunotherapeutic molecule? But in my opinion, you should use in such a way that it brings back the homeostasis and establish the normal system. I would like to stop here and thank people who have been, this is a old slide, to be very honest, and these people have moved on. Mohan is in Pfizer now in the United States. Shiva, these two people did a lot of work on regulatory T cells, and so did Veenu Sharma, and she published a science paper, and these two people did very good job. And then I thank all these people who have been in the lab. They look happy. Thank you very much, and I can take questions. I'll be happy to take questions.
Thank you, Dr. Kaveri. Very nice and very informative talk. And you have given us a very new information about how regulatory T cells can be used for treatment of various diseases. Sir, in one of your slides, you showed, the last slide also, you showed that Treg cells are also known to suppress many immune cells.
Yes.
And I'm sure there could be some clinical trials going on regulatory T cell transplantation. And so what is are they have an adverse effect, or what is the status today?
No, this is a very interesting question, Mambani. This is a fantastic question, because in fact, we have learned a lot on the regulatory T cell function and the Treg immunotherapy, thanks to the transplantation. And to me, a transplantation is as important as autoimmunity in learning more about regulatory T cells. Enormous number of clinical trials are right now going on. Since the talk was more on autoimmunity, I did not want to insist on the transplantation. But my friends right here in Paris, they are working on a variety of, including IVIG, for example. I don't know whether you are aware, IVIG is used as a means for suppressing the HLA, the donor-specific antibodies. And one of the mechanisms that it happens, even in transplantation, is through regulatory T cell function. So answer to your question, yes. In transplantation, regulatory T cells play a crucial role, and this will be exploited in the future to be more specific.
Right. Yeah. Only thing is the dose to be decided, because sometimes they are very potent. Like it is known that one Treg cells can inhibit the differentiation of 100
exactly
to osteoclast. So that may be happening in other cell type also. So that's
yes.
important.
Yeah, you're right. As earlier speakers told, and we have to be and Chandra also, he kept on asking a very relevant question, because how can you translate these observations to clinic? and what stage and what is the guideline that we can know when you are manipulating the immune system. You have to be very careful. You cannot do anything excessively. When you do excessively, you are targeting, you are doing more harm than you are doing good, because immune system is so powerful and sensitive at the same time. So but it will happen. Girdhari answered in one of his answers, what he said is individualized or personalized or tailor-made. The technology is advancing so well that what we are doing is to understand the signatures, the biological markers. See, the moment a patient walks in, imagine in a matter of few hours, if you have a signature of what is the level of activation of different immune components, and then you may be smart enough to design drugs or give appropriate dose to bring down only those activated. But that's a little bit of, right now, the question of Chandrasekhar is so important, of the dosage, what to give, when to give, how much to give. It's very crucial. We have not answered that yet.
Thank you, sir.
Yes.
Yeah.
Any other question?
Yeah, if time is allowed, I think one small query.
Sure.
Now, I mean, see, one of the things which is as the first speaker said, Giri was saying about the plasticity of the T regulatory cells.
Right.
I mean, that gives us an advantage of probably numerically you can little err on either side. So probably the plasticity should take over. But the only problem that comes for us is the prepared cell, is it going to be plastic or is it not going to be? Is there any studies which have been worked on that?
Of course. I mean, this is, the plasticity I underlined, and so did Girdhari. When you design a new set of cells, you better ensure that they are following the same trend of plasticity as the naturally evolved regulatory T cells, or naturally evolved any type of cell, because plasticity is the key. Because imagine if they get stuck, if you get stuck with a particular bunch of cells with an undesired function, you don't want that. You want the plasticity is so crucial, as Girdhari said. The moment their good job is done, yes, they go back to normalcy. They go back to what they are supposed to do. You activate them exactly at the right time, and that's when the plasticity comes into picture. You change your phenotype, change the cytokine profile, do your job. The moment the job is clean, you go back. You go back to you have to ensure that. But we are not quite, there yet, but we will, I'm sure. Yeah. Good. I guess people are tired and they want to, it's already late in the evening on a Saturday.
Just fantastic talk. I think we got actually refreshed, completely refreshed.
Girdhari, but I didn't show so much beautiful data, or you as Sunil, as Rupesh. I mean, you people did a fantastic job. Really, you are the James Bond for me.
And I think to conclude, it was really a wonderful session, though we spent an hour extra of the day than specified. I think, at least me, didn't really feel that there was one more extra hour. I hope Manjula do agree with me.
It's just fantastic. It's so much refreshing that everybody is warm up again.
Yes.
Immune booster.
Immune booster.
Yeah. Fantastic, sir. Listening to experts, it was fantastic.
Nice. So I take this opportunity to thank all the speakers, as well as Mr. Iyengar and all my back staff who really helped in making this work smooth. With this thank, I think I remind everyone that we have the next session tomorrow at 10 o'clock. I welcome all of you to join for tomorrow's Feast with Reference.
No, we should not miss. We should not miss, because there are some more fast and furious kids there. Oh my God, there is Avasti, and then all of them are really fast and furious. James Bonds are coming tomorrow. Yeah.
Right.
Thank you for leveling us up that way.
No, you are. I am your fan.
Thank you, sir. Thank you.
So, thank you. I think we will close the session for the day. Thank you very much.
Thank you.
Namaskar. Let's have a pleasant evening.
Bye-bye.
Namaskar.